So, thanks very much. First of all, I would like to thank the organizers for inviting me to this very nice meeting. The title of my talk is ApoE genotype, oxidative stress, inflammation, and disease risk. And I'm a nutritionist, and we are mainly interested in dietary factors. So we are basically asking the question how diet and dietary factors and genetic factors impact on healthy aging. Currently, dietary recommendations, as in the US, in the UK, and in Germany, are based on age, physiological status, and the prevalence of disease. In the future, we may have also dietary recommendations which may take the genotype into account. And we are calling this concept personalized nutrition or personalized nutrition strategies. So personalized nutrition means that we target subgroups of the population which may be at risk, and we may intervene in this very subgroup with master-tailored dietary strategies. So the genotype we are mainly interested in is the ApoE genotype. And we are basically asking the question if and to what extent the ApoE genotype may affect antioxidant defense mechanisms, chronic inflammation, and how this impacts on the disease risk. In order to study ApoE genotype-diet interactions, we are following up a concept of increasing biological complexity. So most of our studies are done in cultured cells, stably transfected with ApoE3 or ApoE4. We look into targeted gene replacement rodents, and we also confirm some, not all of our data in human intervention or in human cohort studies. And we are using, as many others do, so-called omics platforms. So the most important model we are currently using in our lab is the so-called ApoE3 and ApoE4 targeted gene replacement mouse model. And this is an interesting model where the murine ApoE background is deleted and the human gene is knocked in. Most importantly, the ApoE3 and ApoE4 mice express equal amounts of the two isoproteins. The target tissues we are mainly looking into is the liver. 60% of the ApoE is produced in the liver, the vasculature, and the brain. And we are also, as nutritionists, try to intervene with dietary compounds, and today I will talk about flavonoids and so-called isothiocyanates such as sulforaphane and others. So what is ApoE? Apolipoprotein E is a lipoprotein which is mainly involved in reverse cholesterol transport. So it brings cholesterol from the peripheral tissue back to the liver. But besides its effect in cholesterol transport, it has been turned out recently that it may be also involved in antioxidant defense mechanisms, and it may regulate also inflammatory processes. So ApoE is a polymorphic lipoprotein. 65 up to 70% of us here in the lecture theater are carriers of the ApoE3 allele. 15% of us are carriers of the ApoE2 allele, and 15 up to 20% of us are carriers of the ApoE4 allele, and this is the genotype at risk. The difference between the E3 and the E4 is just an amino acid exchange in position 112, but this very much impacts on the disease risk. The ApoE4 allele is not equally distributed in Europe. It follows a distinct pattern. So the E4 allele, the genotype at risk, is enriched in Northern Europe, and we have a very low allele frequency in the so-called Mediterranean area, where we have a very low incidence of cardiovascular disease. So if we calculate or correlate the latitude with the ApoE4 allele frequency, this follows a distinct pattern. So the genotype at risk is enriched in Northern Europe, and we have a very low allele frequency in Italy or in Spain and increase. And in these countries where we have a high ApoE4 allele frequency, like in the UK, we have a very high risk for cardiovascular disease. The ApoE4 allele is underrepresented in German sanitarians. At the University of Kiel, we have a cohort where a couple of thousand German sanitarians are genotyped, and the ApoE4 genotype is highly underrepresented. It's known since more than 2 decades that ApoE4 is a mortality factor in the elderly, and is one of the most important risk factors for Alzheimer's disease, and it is a very important risk factor for cardiovascular disease. We are trying to understand the role of ApoE genotype in terms of chronic inflammation, NRF2 signaling, its responsiveness to dietary factors, including flavonoids and isothiocyanates, and we have recently also looked into the effect of the ApoE genotype on the vitamin D status. A couple of years, we did a study together with Lester Pecker in California, where we retrospectively genotyped a relatively small sample in terms of the ApoE genotype, and we looked into F2 isoprostanes, which may be a surrogate biomarker for lipid peroxidation. And what we found is a very small increase in F2 isoprostanes, but the differences between the E4 and non-E4 carriers were rather minor. We then looked into cultured cells, and we measured superoxide anion free radical production in ApoE3 and ApoE4 macrophages, and we found that the genotype at risk puts out more reactive oxygen species than the so-called wild type. In our rodents, in our transgenic mice model, we looked into peroxinase mRNA and protein levels in response to the ApoE genotype. And what we found is that the genotype at risk, the ApoE4 genotype, produced lower amounts of peroxinase 1, which is an enzyme which is centrally involved in the detoxification of oxidized LDL. Peroxinase 1 is an enzyme which activity decreases with age. We could show this in a healthy UK cohort, but also in a German cohort. So it's an enzyme which is centrally involved in the aging process. But we don't think that oxidative stress is the main reason for differences in the disease risk between ApoE3 and ApoE4 carriers. We think that modulation of inflammatory processes are at least equally important. So if we go back to our cell culture model and measure the production of pro-inflammatory cytokines, including tumor necrosis factor alpha, we find that the ApoE4 macrophages put out more tumor necrosis factor alpha upon stimulation with LPS. In terms of the anti-inflammatory interleukin 10, we find exactly the opposite. So we have lower interleukin 10 production in the genotype at risk. And this holds true also for other inflammatory cytokines, including interleukin 1 beta, macrophage inflammatory protein 1 alpha, and TNF. We then measured NF-κB transactivation activity in our macrophages, and we found that the genotype at risk has a higher NF-κB transactivation activity as compared to the wild type. So this may explain the higher production of pro-inflammatory cytokines in the genotype at risk. We then did some studies in our macrophages, supplementing them with quercetin, and we found that physiological achievable concentration at 1 micromolar are downregulating inflammatory gene expression. And based on this finding, we went back to the mouse model and measured TNF-alpha production in response to the ApoE genotype. And what we found was that the ApoE4 mice, so the genotype at risk, are less responsive. And we did find a lower nuclear NRF2 level in the genotype at risk as compared to the wild type. So what we basically postulate is that in the genotype at risk, which is the ApoE4 genotype, phase 1 enzymes are enhanced, whereas the phase 2 enzyme response is impaired. And this may very much contribute to the higher disease risk in the ApoE4 genotype. So we are now looking more into detail into the NRF2 signal transduction cascade, and we try to single out nutritional or dietary factors which may correct for the impaired NRF2 response in the ApoE4 genotype. And one compound we are very interested in is allyl isotyrosine, which is a so-called ITC. Normally, sulforaphane is used as a very potent NRF2 inductor, but we have shown that sulforaphane may have also some side effects and it can be cytotoxic. But AITC is less cytotoxic than sulforaphane and more or less equally potent in inducing NRF2-dependent gene expression. This was in cultured cells in fibroblasts. So we then did in vivo studies in our mice, and we fed these mice with AITC. These are wild-type controls, and as you can see here, if you give AITC, you significantly enhance NRF2 nuclear levels in these mice. And this is accompanied by an increase in NRF2-dependent gene expression, but also by an increase in hemoxygenase protein levels. Based on this finding, we then went back to our ApoE3 and ApoE4 mice, and we fed these mice with AITC, and we measured phase 2 enzymes in response to the treatment. Interestingly, the ApoE3 mice, so the wild type, they have higher baseline levels of the phase 2 enzymes, and they are much more responsive in terms of the inducing activity of AITC as compared to the genotype at risk. So much lower levels of phase 2 enzymes in the genotype at risk, and we cannot compensate by giving AITC, or we cannot bring these animals to the same level as the corresponding controls. So another question we are currently asking is it always bad to be a carrier of the ApoE4 allele? And was there any evolutionary pressure that the ApoE4 allele is so much enriched in Northern Europe, but also, for instance, in Africa? Why do ApoE4, as compared to ApoE3 carriers, exhibit a superior vitamin D status and also a superior calcium status? So we measured bile acid production, and this was elevated in the E4s. We measured also genes encoding for proteins important in renal reuptake of vitamin D, and these genes were significantly upregulated in the E4s. We also measured genes encoding proteins involved in calcium absorption, and also we measured calcium absorption, and this was higher again in the ApoE4 carriers. And we measured renal calcium reabsorption, and again, this was higher in the ApoE4 carriers. To summarize, so what I tried to get across is that there are significant differences between the ApoE4 and the ApoE3 genotype. The ApoE4 genotype is the genotype at risk, accompanied with higher cytokine production as far as pro-inflammatory cytokines are concerned, with a higher NF-κB transactivation activity, and with a lower NRF2 phase 2 response. I didn't show you data on vitamin E today, but vitamin E is decreased in the peripheral tissue of E4s, and also peroxinase 1, an enzyme centrally involved in the detoxification of oxidized LDL, is decreased. Biomarkers of oxidative stress, including F2 isoprostanes, are slightly enhanced in the ApoE4 genotype. The phase 1 response is increased, the phase 2 response is decreased, and most interestingly, vitamin D status and calcium status is enhanced in the ApoE4 genotype. So what we can basically say is that the ApoE4 genotype is the genotype at risk, and it is highly associated with aging-related diseases, including cardiovascular disease and Alzheimer's disease. Personalized nutrition strategies may or may not be adapted to counteract the genotype at risk. ApoE4 carriers, and that's the problem, the genotype at risk is less responsive to dietary interventions, and ApoE4 allele carriers may have a higher demand of health beneficial dietary factors, including flavonoids and ITCs. So I would like to thank you for your attention, and also my colleagues and also the funding bodies for financial support. Thanks very much.
Thank you, Gerald, for the great presentation. Now, there are questions.
I enjoyed your talk. Maybe it can be of interest for you that in the European project GER, in which Stefan Schreiber has been involved, we have found that there is an interaction between mitochondrial haplogroups and ApoE carriers. And also the genotype, all these people in Europe about showing that there is this gradient between north and south. So my question is, do you have any data regarding the prevalence of dementia in Northern Europe and in Southern Europe, which could be one of the possible effects? As far as I know, there is not much difference.
Prevalence of what?
Dementia.
I don't know whether dementia is higher in Northern Europe as compared to Southern Europe. I can just say that the ApoE4 genotype is enriched in Northern Europe as compared to Southern Europe. As far as cardiovascular disease is concerned, we have a much higher rate in Northern Europe as compared to Southern Europe, but I don't know about Alzheimer's disease.
ApoE4 is one of the main risk factors for dementia. So if there is a higher prevalence, this could be explained. But as far as I know, there is not much, 3 times more prevalence of dementia in Northern Europe, despite the fact that ApoE4 is considered the main risk factor for dementia. So there must be something protective in northern countries for this effect on brain degeneration.
I agree. But if you, for instance, look into the Japanese population, where we have many sanitarians, we have a relatively low rate of ApoE4. And also within the country, I think that's very striking as well. We have also gradients of the ApoE4 genotype within the country. For instance, within the UK, we have more E4s in Scotland as compared to southern England. And so I think that this may be related to vitamin D, but it may be also related to other factors. But it's not only ApoE4 which predicts the risk of Alzheimer's disease. That goes without saying.